how must it feel to be at the bottom of the world, to be marked and stomped by the galloping herd, to be neglected and abused for all this time? What does our thanks look like to you? You who birthed the food which fills us, stores away the carbon of harm. You who give our walk strength, make the entire ground possible. And yet here we are, eroding you, treating you like dirt, pushing you further into an exhausted bruise. We dump the burden of our cities on your backs, build more of our money temples around the beat of your heart. So perhaps now would be the right time to stop and walk with you, learning more about your way, preserving your wonder, hoisting you up from beneath our feet and into our sights, celebrating the million ways you work, allowing all of human existence to thrive, the tiny miracles you perform each day and each night, like how in a single teaspoon of soil exist more microorganisms than people alive. Want to find out more about the buried treasure in your soil? Come to the Royal Society Summer Science Exhibition.
About 1/4 of all marine species are associated with coral reefs. But also the livelihoods of 500 million people depend directly on functioning coral reefs, and many more benefit from invaluable services such as coastline protection. New drugs have been isolated from reef organisms, and glowing corals have provided innovative tools for biomedical research. Night is the best time to draw the luminous secrets from the corals. By shining blue light on corals, the scientists can induce a process called fluorescence. The blue light of their special underwater torches is absorbed by certain pigments in the coral tissue and then re-emitted as light with red-shifted colors. Researchers and students at the University of Southampton are analyzing this phenomenon in coral reefs in different parts of the world. From the sun-drenched reef crest down to the reef foundation, where the low light levels become limiting for coral growth. Using the experimental coral aquarium in Southampton, the researchers have already succeeded to unravel key functions that these pigments fulfill in the corals. In shallow water, they can act as sunscreens for the symbiotic algae that live in the tissue of the coral animal. In deep water that is naturally dominated by blue light, the transformation of light color by the fluorescent pigments helps the algal partners of the coral to make better use of the light for their photosynthesis. Due to the amazing optical properties of the fluorescent coral pigments, they can be used to study the functioning of living cells and tissues.
Surprisingly, only a single gene is required to make these fluorescent molecules, so it's relatively easy to introduce them in cells under study. Their glow can then be used to visualize cellular processes under the fluorescence microscope. The application of fluorescent proteins as imaging tools in medical research helps us understand how diseases such as cancer, Alzheimer's or HIV affect the functioning of the cells within our bodies.
The researchers at Southampton are screening the reefs for novel fluorescent pigments that allow new imaging application. One of their coral pigments that can change its color from green to red helped with the invention of a microscopy technique that allows us to study cells with super resolution. Once promising candidates are found, the genes required to produce the protein pigments responsible for the glow are then isolated and analyzed in detail in the molecular laboratory. Novel fluorescent pigments are subsequently tested in imaging applications in collaboration with medical researchers at the University Hospital Southampton and colleagues in research institutions around the globe. For this purpose, DNAs that contain the information for the production of a protein of interest and for the glowing protein are modified by molecular biological techniques and are fused together to build an artificial gene. When this gene is introduced into a cell, the two proteins form a tandem in which the glowing tag allows us to follow the invisible partner on its way through the living cell. The diversity of coral colours allows us to label different cellular building blocks and follow complex biological processes such as cell division. Students at the University of Southampton follow the glow of newly discovered pigments in their practical classes when they learn about biochemical techniques. However, the imaging power of fluorescence may not only be helpful in medical applications. The researchers want to use it to monitor the health of the corals themselves. Rising seawater temperatures and deteriorating water quality threaten coral reefs.
So fluorescent pigments in coral skeleton help us date and analyse past periods of stress using isotopic and geochemical techniques. The pigments we're looking at, though, they're different to the ones that are in living coral tissue, but we can still use fluorescence to detect their presence. And since they're only incorporated in certain times of the year, they form a banding in the coral skeleton, and we can use that banding to date the coral in the same way that you can use tree rings to date a tree.
The coral tissue itself responds in very distinct ways to different forms of stress, such as changes in light and temperature levels, wounding, or the presence of parasites. When the causes for these colour changes are understood, they may help to monitor reef health.
Fluorescence is extremely powerful when it comes to imaging, so we apply the knowledge that we gain from evolving coral pigments into microscopic tools to develop monitoring approaches for coral reefs. For example, fluorescence can be used to spot and count coral recruits on reefs that are recovering from episodes of stress.
The Southampton researchers hope that they can contribute with their efforts to save coral reef ecosystems for the future.
When we're faced with an engineering challenge, chances are nature has already found an elegant solution. Millions of years of evolutionary trial and error has led to some ingenious adaptations and mechanisms. We can use this as inspiration, leading to remarkable innovations in science and engineering, what we call biomimetics. And sometimes we need look no further than our own back garden. Scientists from the Natural History Museum in London and Imperial College London have come together to deliver 3 of the most exciting innovations in biomimetics. Medical glues have been used to close wounds for decades, but they're often rigid and brittle and can't be used for internal injuries because they can be toxic to cells and fail to stick to tissue in wet conditions when surrounded by blood. Slimy slugs have inspired a new kind of adhesive that could transform medicine. The dusky Aryan slug creates an extremely sticky mucus as a defence mechanism against predators. Scientists have engineered a unique bioglue that mimics key features of this slug mucus. When tested, this biocompatible medical adhesive remains incredibly sticky, even in wet environments, and can undergo movement and physical stress and strain without leaking or breaking. This sticky, strong and flexible glue could be the most successful future alternative to stitches and sutures. Dragonflies are aerial acrobats. They can control the angle and speed of each of their 4 wings independently, allowing them to fly in any direction, hover, and perform astonishing manoeuvres. The dragonfly wing is a live structure containing hundreds of neurons which provide constant sensory feedback. This is what enables dragonflies to control their flight with such speed and precision. Using specimens and the micro-CT scanner at the Natural History Museum to study the wing structure closely, scientists have an ambitious goal to replicate a similar kind of feedback system and make aircraft wings live too. This could mean designing an aircraft with wings that could sense and respond to changing conditions automatically and independently, which could revolutionise flight. Flies are constantly on the move. They have highly efficient visual processing systems, along with other specialised sense organs on the body, which helps them remain balanced and stabilise their gaze as they navigate their environment. By using cutting-edge technology at the museum, scientists are able to look at the anatomy of various fly species in minute detail, right down to the muscles and joints. They can also monitor the electrical activity the fly brain generates in response to visual motion, activity that's key to controlling head and body orientation when the animal's on the move. By mimicking this efficient system in flies, bioengineers at Imperial are opening doors to the possibility of robotic systems like drones that have the right sensors on board to perform vital tasks autonomously.
So it's been really great working with Adam. Normally the collections here get used for studies such as taxonomy and systematics, evolution, ecology work. So seeing the collections used for new cutting-edge science, things like bioengineering, has been really amazing.
Yeah, similarly, it's been fantastic having access to the collections at the museum for myself and my colleagues at Imperial. Often we find that when we look to nature for solutions to engineering problems in bioengineering, often nature's found the perfect solution already, and we just have to try and mimic that as best we can.
This is one of the world's oldest known fishhooks. It was made from a seashell by humans living some 23,000 years ago. Humans are extraordinary tool users. We completely rely on tools during our daily lives, and we've developed technologies of astonishing complexity. Why don't more animals use tools? For every 10,000 species, there is only one that is an accomplished tool user. On a remote island in the South Pacific lives a bird that has baffled scientists with its remarkable tool-using skills. The New Caledonian crow. These birds use sticks to winkle insects from hiding places, and are even capable of crafting tools from raw materials to suit their particular needs. In fact, the New Caledonian crow is the only species beside humans that has developed tools with that simple but incredibly useful fisherman's device, the hook. We wondered whether there are any other crows that might be able to use tools. We began to search for species that look similar to the New Caledonian crow, whose unusually straight bill is perfectly adapted for wielding stick tools. It seemed to us that the Hawaiian crow, one of the world's rarest birds, is a particularly promising candidate. Our hunch was right. We've recently discovered that Hawaiian crows are incredibly good at using foraging tools and deserve to join the exclusive Tool Users Club. And it may be no coincidence that this species also lives on a remote tropical island, just like the famous New Caledonian crow. This year, at the Royal Society Summer Science Exhibition, you can find out more about our discovery, and you can investigate for yourself how tool-using crows can help us understand humans' remarkable tool-making journey, from fishhooks to space shuttles, in under 1,000 generations.